Good morning. It's March 18th, and this is your daily brief in science. Here's everything you need to know. A daytime meteor over northeast Ohio produced a loud sonic boom heard as far west as Norwalk and as far east as Pennsylvania, with multiple observers confirming the event. NASA estimates the object was a small asteroid about 6 feet in diameter and weighing roughly 7 tons, a standard meteoroid classification. The fireball was seen near Medina, rising brightly through the atmosphere, and satellite imagery along with lightning mapping data support the meteor hypothesis. Officials and science journalists note the scientific significance and rarity of daytime meteor events, while stating data is still being reviewed and more information may emerge. Authorities reported no injuries or property damage and described the incident as a developing news story with ongoing updates. Experts say small objects commonly enter Earth's atmosphere and burn up, with little risk of widespread damage. Residents are advised to look for small meteorite fragments, dents in cars, or tiny holes on outdoor objects as potential evidence of meteorite impacts. It remains unknown whether any fragments landed. Recovery could be possible if a meteorite reached the ground, with higher chances in accessible areas. The exact size and trajectory aren't confirmed, but officials emphasize the meteor explanation based on current data. AMS provides an outlook for meteor activity in the week, framing the event within ongoing near-term activity without indicating dangerous conditions. Medina County is a focus for potential meteorite recovery, with experts noting that tiny black rocks could be found. The incident highlights that daytime fireballs with audible booms are rare but noteworthy for analyses of size, speed, and trajectory. A UCLA-led team has created an implantable device that acts as a charging station to reinvigorate CAR-NKT immune cells near tumors, boosting durability and anti-cancer activity. The platform uses biomimetic microparticles to provide activation signals via TCR antigen and to deliver IL-15, supporting CAR-NKT proliferation so reactivated cells can circulate systemically and target cancers beyond the implant site. In preclinical studies, this approach improved durability and anti-tumor activity of CAR-NKT cells in solid tumors and systemic blood cancers, concentrating signals at the implant to reduce systemic toxicities. Biocompatibility was demonstrated with minimal adverse effects in animals, and signaling is localized to avoid cytokine release syndrome, suggesting potential for combination with other therapies. Key researchers include Song Li and Lily Yang at UCLA, with Yan Rudili as first author, along with Haochen Nan, Zheyang Liu, Yucheng Yang, Xinyuan Shen, Ying Fang, Yichen Zhu, Yuning Chen, Zibai Liu, Zhengyao Shao, Bo Zhang, Zheng Xiai, and Enbo Zhu. The work is affiliated with UCLA's Broad Center for Regenerative Medicine, Johnson Comprehensive Cancer Center, Molecular Biology Institute, Parker Institute for Cancer Immunotherapy, and Goodman-Luskin Microbiome Center. The research outlines a broader vision for modular, localized immunotherapy platforms that could be adapted to other cell therapies and cancer types, with ongoing work to optimize efficacy, durability, and safety. Published in Nature Biomedical Engineering, the study underscores biocompatibility, optimized signal strength, growth-supporting protein release, and maintaining localized signaling to minimize systemic toxicity. The approach aims to maintain localized immune stimulation to minimize systemic side effects while sustaining immune cell activity, potency, and long-term memory formation. Researchers emphasize balancing stimulation to avoid immune cell exhaustion and focusing signals locally to broaden applicability across cancers. Funding comes from the California Institute for Regenerative Medicine, NIH, DoD, and UCLA, highlighting strong public and academic support for this immunotherapy direction. Nature Biomedical Engineering published the study, noting validations in human melanoma and lymphoma samples and mouse models, underscoring translational potential for solid and blood cancers. A Nature Geoscience study reanalyzed 2,315 seawater samples from the North Pacific, Baltic Sea, South Africa coast, estuaries, and reefs to map the distribution of human-made chemicals in the ocean. Researchers found widespread contamination by anthropogenic chemicals, including industrial compounds and everyday personal care products, across Pacific waters and other sampled regions. The lead author says understanding and mitigating the long-term impacts of chemical intrusion on marine ecosystems is urgent. Industrial chemicals tend to decline with distance from land, but plastic-derived compounds persist far offshore, contributing to the marine carbon pool, alongside micro and nanoplastics. The findings provide a framework for future toxicology studies and policy decisions on chemical waste management, prioritizing which molecules to monitor. There are concerns about potential bioaccumulation of synthetic substances in the marine food web, with implications for biodiversity and communities relying on ocean resources. The study notes potential implications for ocean health and carbon cycling, but exact effects require further experiments. Researchers call for more lab work to understand how industrial pollutants integrate into global carbon cycling and affect microbial communities from plankton to whales. Chemical patterns show industrial chemicals are widespread, while pharmaceuticals and pesticides are more localized to coastal waters, indicating significant coastal infiltration. Using non-target detection, researchers identified a broad suite of pollutants, including beta blockers, antidepressants, antibiotics, cocaine, methamphetamine, DEET, and atrazine, with coastal samples near Puerto Rico showing pollutants making up to about 1/5 of dissolved organic matter. Personal care product-derived compounds are among the most pervasive pollutants, especially near coastlines, where they can comprise up to 20% of dissolved organic matter in some waters. The study identifies 248 distinct synthetic chemicals linked to human activity, offering a detailed inventory of how human production alters dissolved organic matter in the sea. A joint HK-UUSTB team led by Professor Yang Liu and Professor Cheng-Ming Li has produced a freestanding ultra-hard diamond wafer up to 5 inches in diameter and 3 millimeters thick, achieving a Vickers hardness over 200 GPa. This marks the first time inch-scale size is paired with ultra-high hardness in diamond. High-resolution TEM shows a dense network of three-dimensional interlocked stacking faults, reaching up to 4.3 times 10 to the 9th cm-2, which suppress dislocation motion. Nitrogen reduces stacking fault formation energy, promoting their growth according to EELS and first principles calculations. The ultra-hard wafer is positioned for extreme environment electronics, advanced manufacturing, and semiconductor thermal management, with potential applications in MEMS, high-power and high-frequency chip cooling, and advanced packaging. A customized MPCDV system and a high-frequency pulsed nitrogen doping strategy create a rapidly switching non-equilibrium growth environment during diamond deposition, enabling the observed microstructure. Mechanical tests report a Vickers hardness of 208.3 GPa and wear resistance about 7 times higher than typical polycrystalline diamond substrates, along with the ability to machine high-quality single-crystalline diamond surfaces, signaling strong machining potential. The study is published in Nature Communications under the title Inch-Scale Ultra-Hard Diamond Wafer with 200 GPa hardness via high-frequency pulsed local non-equilibrium growth. Researchers foresee controllable modulation of microstructure and band structure as essential for next-generation diamond-based microelectronics and optoelectronics, with the wafer serving as a platform for diamond MEMS and nanostructures. A landmark genetic study maps the overlap across 14 psychiatric conditions, showing substantial shared risk that spans internalizing disorders, substance use issues, ADHD, and related conditions. This largest and most comprehensive analysis to date draws from over 6 million individuals, revealing a broad pleiotropic genetic architecture that could reshape how we understand biology and treatment. Researchers identify 428 genetic variants linked to multiple disorders and 101 chromosomal regions with high concentrations of shared variants, indicating widespread overlap. 5 disorder groups emerge from the data: compulsive disorders, internalizing disorders such as depression, anxiety, and post-traumatic stress disorder, neurodevelopmental conditions, schizophrenia and bipolar disorder, and substance use disorders, with clear crosstalk between these clusters. Biological patterns align with the genetic findings, as internalizing disorders show enrichment for oligodendrocyte-related genes, while schizophrenia and bipolar disorder link to excitatory neuron-related genes. The study notes that ADHD and autism spectrum disorder share strong genetic correlations, and later diagnosed autism with ADHD and other conditions is highlighted by a 2025 study. Co-occurring conditions are common, with about half of people with one disorder receiving a second or third diagnosis. Anxiety and depression frequently accompany ADHD, underscoring real-world clinical complexity. The researchers frame these overlaps as a shared genetic scaffold that could inform more integrated screening and intervention strategies, guiding future work toward tailored approaches based on genetic risk profiles. Cited sources include Nature and prior DSM-based prevalence research, placing the findings within established scientific literature. Overall, the work points to a unified biological framework across diverse psychiatric conditions and encourages more comprehensive approaches to diagnosis and treatment. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.